for a process called polymerase chain reaction, for which Kary Mullis received the Nobel Prize. It requires that you have an oligonucleotide, a primer, a single-stranded DNA, and it also requires temperature-stable DNA polymerase, because otherwise DNA polymerase gets denatured at high temperature, which you have to use to separate a double-stranded DNA. So if we take a double-stranded DNA and we denature these two DNAs, we can add primers that correspond to the 3' ends of each of the longer DNAs. The primers run 5' to 3' to the 3' to 5' of the DNA. We can elongate using polymerase and get 2 copies of that DNA by copying each strand of the DNA with a primer. We can then denature for a second cycle, add the primers are already there, the polymerase is already there, and so you denature, you allow it to reanneal with the primers, and now you have 4 strands of DNA. So this is a way to amplify DNA sequences using this temperature-stable DNA polymerase that allows denaturation at high temperature, annealing at low temperature. DNA polymerase stays stable through the cycles of temperature. This allows amplifying any DNA sequence. Complementary DNA derived from a sequence of messenger RNA, genomic DNA, any DNA sequence can be amplified as long as you have a primer. And that amplification is exponential. That is, first cycle you get 2 copies, and then you get twice that, 4 in the second cycle, 8 in the third, 16 in the fourth, and after 30 cycles you have exponential expansion and about 1 billion copies of the same DNA sequence. So this allows experiments with sequences that are very hard to isolate, that are very rare. And so if you explore the literature, for example, on looking at synthesis from Neanderthal DNA, that is possible because of this kind of amplification, which has to be done carefully under very controlled conditions. So what are the kinds of applications for this other than amplifying DNAs that we want to look at? Well, it turns out there are various ways that you can carry out PCR. You can do quantitative PCR, which uses a fluorescently labeled dye on the oligonucleotide to track amplification in real time. When that dye gets into double-stranded DNA, you can separate it from the primer so you can see how much has actually been amplified. You can actually use an enzyme in the first step that makes a complementary DNA from an RNA sequence. And then that RNA sequence can be amplified by using the initial RNA, the DNA oligos in the first step, and then normal PCR in each subsequent step. And there are a whole variety of variations that are all based on using an oligonucleotide, a template, and a temperature-stable DNA polymerase.